Thank you, JD. Thanks everyone for being here tonight. It's a great privilege to be here. I'm glad to see so many people in the audience. Just to start off, I want to show you an image of a frog. So how many legs does a frog normally have? Yeah, probably not that many. Well, this is what I study as a biologist. I'm a herpetologist. I'm really interested in complexity of development in complex ecosystems. This is really different than the malformed embryos we were just looking at with Ben's fish. This is actually a physical deformity. It's caused by mechanical disruption from a parasite, though the parasite seems to be increasing, or at least having a greater impact in some populations of frogs now. It's considered an emerging disease in North America, though I just spent most of the summer in the Netherlands and found one of the hottest deformed frog spots I've ever seen. One of the hottest in the literature where this parasite's not supposed to be. But anyway, this isn't a frog talk. If you're interested in frogs, please talk to me after this talk. So as JD was saying, I kind of wear many hats. I'm an artist, biologist. As I said, I love frogs. Again, that niche is I'm a herpetologist, so I'm looking at anurine amphibians, frogs, and toads. I spend a lot of time in the lab, most of my time in the field, but I'm really, really interested in bringing people along in that process. I believe really strongly in citizen science, the idea that biology can become participatory, that it can become democratic, and it's a good way to get people interested in their local landscapes. And the local organisms they're sharing eco regions with. So I work with the public a lot, I work with school groups, I do educational programs, and then it all somehow inspires art. And the art kind of inspires more science and more programs. It's a whole kind of system. A specific focus, as you can imagine, is bioindicator species. So I'm really interested in this idea of what, as far as the artwork goes and as far as a lot of the research goes and the programming, kind of what it means at this point. That one of our guests mentioned the Anthropocene. Ed brought this up when he was talking about his project, but what that means, how that's affecting not just population dynamics of other organisms or the functioning of ecosystems, literally how it's shaping organisms and shaping evolution and natural history and potentially, if not already, shaping our own species. So this is an example of one of the public field trips. I call these eco actions. So it's literally training people to be volunteers to help me collect data. So I try to work with all age groups to different degrees, and then also setting up open laboratories. So laboratory settings where real science is done, so it's primary research, but inviting the public in, say, 3 days a week to come in and look at the process and have a conversation about the experiments that are going on there. So I like this kind of dialogue. And with the field work, it's so glamorous. Just having this ability to be out in the mud, getting eaten alive by mosquitoes, and then you can do contests like who can keep a leech on their face the longest. I'm usually the only participant. But the point really is that it's about trying to connect people back to their natural environment in a time of rapid ecological change. What does it mean to be a human being at this moment in history? In the Anthropocene, where so much loss of biodiversity is happening, so many changes are happening to ecosystems. How do we reconnect people? And how do we inform them? You know, this is one of the great things about science. I mean, it's a tool for understanding the world around us. So is art. How do we share that with a larger community? So more in the context of the pieces upstairs, hopefully some of you saw them. If not, we'll see them together in a few minutes. They're relating to the Deepwater Horizon oil spill. So as I said, I'm a herpetologist. I'm not a marine biologist. I'm not a toxicologist. I'm no expert on oil spills, anything like that. But when I was hearing about what was happening in the Gulf of Mexico, I was concerned not as an artist or a scientist, but as a citizen of this planet. I wanted to know what was going on. And so I started going down to the Gulf states, meeting people, meeting researchers, trying to talk to different community members, going to these beaches myself, collecting dead fish, collecting samples, teaming up with people as much as I could, and then wanted to respond somehow. So the other thing is I was given the opportunity to have a show in 2012 in New York. So it was going to be my first big show with a new gallery, And I approached them about the idea of doing something with the Deepwater Horizon oil spill, because I'd been working on this whole body of work. And the response, not so much from the dealer, but from most people that I was talking to, is they were like, well, that's fixed, right? I mean, it's like, that happened 2 years ago. It's all repaired now, or it's gone. And it's like, even inside the environmental community in New York, I found that not many people, it was like off radar. It was out of the mainstream media, like it was somehow gone. So it was all cleaned up, right? I mean, this is, 2 years ago. Well, I think it might have something to do with, it was really effective. It was $50 million that was spent in the first 6 to 8 months on public relations by BP to spread this message somehow that they were fixing everything. So I don't want to get too political about that, but to hold this in context, $50 million, right? Here we are at the National Academy of the Sciences asking for donations, but $50 million just for PR in 6 months, right? Imagine how much environmental restoration could be done for that. How many environmental programs? So I don't know, these numbers bug me, this kind of something when there's so many people in the sciences in studying loss of biodiversity that have problems getting a $10,000 grant to do research on a species that's endangered. Anyhow, so just some of the facts. I won't go into too much detail. Kim did a great job covering all this anyway. Just a few numbers I wanted to throw out. I noticed some of our numbers don't match up. I don't generate any of these numbers, I just find them. So most of them are from peer-reviewed sources. Like when we go upstairs, you'll see there's a journal, there's an appendix that go to different bodies of work. I try to gather all the data together in one place, or all the studies together, and then people can make their own decisions about what those papers suggest. But the one thing that I wanted to mention is this idea of chemical dispersants, and the fact that these dispersants, which molecularly, from what I understand, again, I'm not a chemist, they bind with oil. Structurally, they break it up, but they make it heavier. So a lot of the oil ended up sinking. So in 2012, 2014, in a few weeks, 2015, that heavy oil in smaller molecules went somewhere. Well, a report from 2013 said about half the oil is still in the Gulf. That's a congressional report. Hopefully that's pretty accurate. Secondly, I mean, again, as Kim was talking about, you never can quite tell. I mean, this is such a huge environmental system that's constantly changing. So where did that oil go? The other thing that some studies have shown is by using dispersants, it made it 52% more toxic to marine life. Secondly, it inhibited the growth of bacteria that would naturally eat the oil. So the question is, why in the world would you use dispersants that are making it heavier, that are slowing down the process? Anyway, it's a different debate that we can get into later, but just these kind of facts really started inspiring me to want to bring this message again that the oil spill wasn't over, and the fact that research needs to continue to the public. So one of the ways that went about doing that is really looking at the food chain. So the complexity of the Gulf food web, I mean, it's incredibly complicated. There's thousands of different species mixing together. Who knows what's happening when those chemicals are entering those different bodies of the ecosystem. A couple of papers, besides Ben's work with malformations, a paper that was published recently from Proceedings of the National Academy of the Sciences, led by a guy named John N. Cardona, also showed that it wasn't just happening in small fish like killifish, they're finding it in amberjack and tuna. Another study from a few months before from Barbara Block showed developmental malformations in the heart, causing cardiac arrest in bluefin tuna. So, obviously the impact on populations could be quite broad. And so somehow I wanted to respond to that. And so thinking of that idea of the food chain, 4 of us put together, it was 2 marine biologists, an ecologist, and myself, and one of my art students, spent about 2 years collecting specimens, sourcing them out, trying to put them together in a complicated sketch. This is only a tiny sketch. What you see upstairs, it's a fraction of the biodiversity in the Gulf of Mexico. So literally what's happening is it was never meant as a visualization. Each jar I curate So literally trying to look at each one as though it's a separate artwork, but put them together to kind of represent that complexity, and also to represent the way that, you know, environmental stressors might be a part of it. This is oil that was collected a few weeks, 2012. So this was like, I started to install this in March. I think this is from February 2012 from Grand Isle, Louisiana. So the specimens we were collecting, some of them came from, these came from Gulf Restoration Network. There's some shrimp downstairs with deformities that, or malformations probably, that came from a guy in Alabama, a shrimper. So lots of people were sending lots of different specimens. Jack Rudlow, he runs a marine lab in Florida. We would go out and get deep sea creatures as part of the installation. And this is one of the shrimp from Alabama. So with what appears to be oil staining. So again, this wasn't science, but it was informed by science. And it wasn't meant as a scientific visualization. It was actually meant more as a kind of aesthetic response, an expression of that complexity, just to kind of start to look at this really complicated ecosystem. It's only, you know, there's 26,000 specimens in the piece. Yes, we had to sit through. You'll see some of the jars. There might be like 3,000 tiny little sea snails inside. It would take about 2 days for us to go through, but we wanted to like make sure that everything was accounted for. So there's about 26,000 specimens in the piece, but that's, you know, out of 370 plus species, it only represents like less than 3%, like 2.4% of the known biodiversity in the Gulf. So literally a sketch. There have been some other bodies of work. Oh, I should also say, but we can talk about it when we're up there, there are also empty jars to represent species that are potentially in decline or will go extinct in the future. And it's this whole idea of what happens when you're taking things out of an ecosystem. I mean, when we look at marine ecosystems around the world, all the fisheries on the planet at this point are in decline. Sadly, that's just statistically the fact. Which I'll talk about some of those numbers in a few minutes, but a related study that Ben and Jack and I worked on together. It's a tiny little study. We looked at about 700 grass shrimp, those collected in Louisiana that were exposed to Deepwater Horizon influence, and those collected from seemingly more pristine habitats in Florida. Same species. These are these tiny little palomades. I think I don't know if they're popcorn shrimp. Do you know if palomade species are popcorn shrimp? They are? Okay. So this is what Popeye sells. Anyway, so looking at that idea of malformation, I wanted to look at a smaller species because these had been used in the 1970s and 80s as a kind of toxicology experiment. So I was looking at them the same way that I look at frogs, just from the exterior, not the interior, just looking for developmental abnormalities on the outside. So when I went through and just with my students and I, looking at just physical abnormalities, it was shocking the difference. I mean, those that were collected from more pristine habitats had about 7.6% injury or some kind of abnormality. Those collected from oil spill sites were upwards of 80%, including things like this. Anybody guess if a shrimp should have legs growing out of its back? Right, I mean, this is a little different. This is totally different developmentally from the amphibian work that I deal with. I don't have an explanation for this. Anyway, so tissue growing on the side. So just this alone, I mean, this was just a pilot study that we did on our own, but it's by getting this information out there, hopefully like encouraging people to realize that more research needs to be done. Research needs to continue, that this is a long-term investigation, long-term remediation. And then more recently, some of the prints in the exhibition. This is part of a project to fund or to help raise money for Gulf Restoration Network. So they're prints of cleared and stained specimens. Let me explain what clearing and staining is. Oh yeah, I should have done that with the frog at first. Okay, so it looks like an X-ray, but it's not an X-ray at all. It's a chemical process. You take a preserved specimen, so something that's been preserved in formalin, you scan and eviscerate like you're dissecting, and you put them in a series of dyes which adhere to specific tissues. So the blue is for cartilage, and the red is for calcified tissue or bone, and then you use a series of chemicals to make them transparent. So literally the finished specimen looks like this. It's like a brightly colored three-dimensional X-ray. So, and then I scan them. And so this is just, out of like, I don't know how many thousands of specimens that we collected going down over the past like few years, but I just chose 10 that looked really nice when they were cleared and stained, and I did these as pilot studies to see if I could see any kind of like weird morphology in any of the fish, which I did not, but I still found them like really beautiful images. So they became a new addition. So there are some of them upstairs we can look at. Triggerfish. And so the idea is that these are kind of apparitions, but kind of there and not there, just kind of a reminder of the loss of the biodiversity, not just in the Gulf of Mexico, but specifically to that area following the oil spill. Greenhouse gate. So, okay, now, try not to read too many numbers at this point, but I guess one of the big points of the Ghost of the Gulf piece, the collapse piece, all of my work is dealing with this like mass genocide, for lack of a better term, of biodiversity that's happening at this moment in history, in our lifetimes, right? I mean, we're like destroying so much of other nature. It's remarkable. This new report, 52% since 1970, so that's in most of our, you know, almost all of our lives here. Probably some people are a little younger than me, hopefully for you. So 52%, right? I mean, this is a huge amount of decline. So it's like other numbers by a report from 2010. I mean, in amphibians, we're looking at about 42% of known species of amphibians that are in decline now. Most of those, again, in 30, 40, 50 years. The numbers are just these sharp declines. It's other numbers, 12% of birds, 22% of mammals, but basically an extinction rate that's about 200 times higher than the normal background level. So, and then again, it's not just happening with vertebrates or terrestrial vertebrates or marine or freshwater species, but also in arthropods. I mean, traditionally people thought that arthropods they could survive anything, right? Nobody really thought to look at arthropod populations because they're so adaptable. Well, now some studies, that study from, that was published in Proceedings of National Academy of the Sciences in 2011 by a guy named Cameron and his team showed that 96% of some not European bee populations, but honeybee populations, native honeybee populations, and bumblebees had declined that much in about 15 years. There's also, everybody's heard of monarch die-off. So it's like we realize so many of these things are happening around us, and I'm sure most of us are familiar with it, but when you look at the stats, I mean, we have to start asking this question, you know, how can we save biodiversity, and who can do it? And it's all of us. I mean, we have to do it. We just have to make the transformation to make it happen. I think collectively, we're a creative species. We have these incredibly complicated environmental, large-scale problems, but we're also remarkable. I mean, look at the art that we do. Look at the science. Look at the music. Look at the food. Look at each and every one of us. I mean, it's every action that we take. It's how we choose to get here. It's whether we choose to drive, whether we choose not to drive, what we choose to eat. Every time you go to the grocery store or dinner, you're voting. So it's like individual decisions. What you're wearing, how you're living. Like, I think it's all reliant on all of us, and we're creative, and we can do it. We just have to do it, so one way or another. So thank you all very much.